Okay, excellent. So today we will continue to talk about some basic calculations and the basic laws, physical laws. Okay, and then for the announcement, I have two updates. Both actually two papers, two reading papers. I put it into the Canvas now, so you can find them in the Canvas shared folder. Because some of you after last class actually talked with me and want to learn more about the persistent length and about the mechanical properties of the cytoskeleton. So to answer those questions, I add two book chapters into the Canvas folders. So then you can you feel free to read those materials after the class. Okay. If you have any questions regarding to any concepts there, tell me immediately, and then I can schedule an individual meeting with you to explain the concepts. If we have time in the class, I can talk more about those important and very interesting cytoskeleton mechanics for you. Okay, that's no problem. Only thing is that our time in the class is very limited, and we have invited several external speakers to talk. Right now, actually, I wanted to keep the number of the invited speaker to be about 5. Right now, we already have 4. Okay, so I want to make sure we have enough time to talk, to share the materials during the class. So, but if you wanted to, even in the limited time, I still want to talk more with you and want to share more knowledge with you. So if you want, we can schedule meeting outside of the class during the office hour or other time. We can still discuss. Okay, so you are always welcome to talk with me anytime. Okay, so here are 2 reading materials uploaded. So feel free to read them. And also we will have invited a lecture this Friday. So we will join Dr. Huang's lecture. So this will be a very interesting talk. He actually combined the mechanics and the biology together on the membrane, on the cell membrane. That's a very interesting topics, and it has a fundamental impact in the drug delivery. So feel free to ask him questions, challenging him any interesting questions during the talk. Okay. For the survey, many of you actually sent me very good survey and told me that you are learning well and you have gained some new understanding. That's great. That's great. I'm happy to hear that. And for rest of you, if you never send me the survey, please feel free to send me whenever you feel comfortable. Okay. So this is a way for us to communicate to make sure that the class is doing well for you, and then let me know anything can be further improved for you. Okay, so that's actually important for you to enhance your learning experience. So feel free to send me a survey anytime. Okay, good. So let's continue our last discussion. So in last lecture, let me see. Okay, can you see the new slides? The cytoskeleton slides, can you guys see that?
Yeah, we can.
Yeah, excellent, good. So in last lecture, we asked a question, a coffee problem, that when the cytoskeleton in the cell generate tension force, especially the actin, they generate tension force. How can we measure those force in the cell? We ask this question. So, for example, here this is a model of the cell that there's the actin filament. They generate tension inside the cell, and then those tension force actually is balanced by the compression force in the microtubule or in the extracellular matrix through those adhesion proteins, those yellow proteins, right? So those actin filament, they generate tension, and then that tension force actually is helping the cell to maintain the stable shape, and also the tension force can act like a signaling pathway. So if you perturb one end of the actin, then because the actin is in the tensed mode, then the force will distribute the vibration in one end will be propagating to the other end, and then let the cell knows there is a force applied to the filament. Okay, so those tension force in the cell is very useful, and it will be important to quantify each different actin filament inside the cell. What is the tension within that? So in last lecture, Daniel, when I asked this question, Daniel raised a point to measure the whole cell force. Okay, now let's go one step further. We want to measure each single filament, actin filament, what's their force within this filament. Okay, so that's the question we asked in last lecture: how to measure tension in single actin cytoskeleton. Now I wonder, after one weekend, whether you have think about this further, whether you have any ideas to measure that. Now feel free to talk, to contribute your idea.
I remember.
Go ahead, Cole.
Oh, sorry. I was just gonna say that I remember from last week, the last idea I brought up was to somehow grab the ends of the actin filament and stretch it, and then apply a perpendicular force to test the displacement, and then use that to figure out the tension. But as far as I thought a little bit about an actual method to do it, and it would require some form of nanotechnology to actually grab those actin filaments and stretch them out.
Grab the actin filament to stretch them out. That's quite difficult. Actually, what we want is a method that will not perturb the cell. Okay, so what you are talking about is you stretch the actin filament, you stretch it. Then what if that stretch point is connected to the adhesion proteins to the membrane and then to the environment? Then you actually perturb the cell behavior, right? And also,
right.
you didn't explain very clearly why you want to stretch the actin, and you also said you will apply some force perpendicular to the actin. Can you explain that idea more clearly?
Sure. So
In theory, how does that work?
The only way for this to work would be if there is some way to extract a single actin filament from the cell. Although I'm not sure if that would be detrimental to the actin. We would need to somehow get it out of the cell without damaging it. Oh wait, no, but we want to know what the tension is in the cytoskeleton while it's in the cell, don't we?
Exactly, within the cell.
Oh, okay.
So you
Then
don't
that makes it
perturb.
a lot more difficult.
Yeah.
Okay, interesting.
Actually,
Because my point with the force was simply that if you I was thinking of it kind of like a physics problem where if you apply the same force enough times and measure the displacement, you could slowly figure out you could determine a physical equation that relates the tension in the cytoskeleton to the force you apply and the perpendicular displacement. I don't know exactly what that equation would look like personally, but I imagine there has to be something like that governing how it reacts.
I think you can do that. I think it's just trigonometry. If you know what the force is, as long as the ends stay fixed, if you have some displacement in the middle, I think you can just like measure the angle of the displacement. And I think you can just use trigonometry to determine the
Right.
tension in
I
the
think you're
filament.
I think you're right, but unfortunately that still doesn't solve the problem of how do you actually do that in a cell without interrupting the other parts Maybe if there were a way to like grab and hold each end of the actin cytoskeleton,
I
wherever
think
they
even if they're
exist.
not fixed, things like optical tweezers could potentially make that possible.
Yeah.
Interesting.
Let's don't there here becomes two questions. Actually, I ask one question. So my question is, if you have a string, if a string like this, okay, both top end, do you guys see my joining here?
Yes.
Yes.
So we have a string here. Now top and bottom is anchored. Now this string actually is not just standing there. This string actually is pulled. So there's a tension T inside the string. Now what I want to know is how do you measure this tension already within the string? And then the two ends of the string you cannot change. So this string is actually mimicking a filament, actin filament inside the cell. That is my question. And you said you raised another question, that is how to do that, how to apply force in the cell. How to apply force in the cell. That's a technical issue that it can be solved. Okay, so right now we are talking about the theoretical issue. If you have a string here, they have a tension inside, initial tension inside. How can you measure that tension? Last time I gave a hint, that is apply a force horizontally, means perpendicularly to the string. But how does that work? If I apply force here, so let's see. If I apply a force here, how can I know this tension? Let's call it a T equal to T0, right? Let me see. Oops. Here is actually the tool. Hold on. That one actually is the tool that you can apply force. But right now, let's fix the theoretical question first.
So I
T0.
believe,
Mhm.
I believe if you deform it and you have a constant force, so if you draw the string lagging to the opposite direction you're applying the force, then you can just break down the tension into X and Y vectors and balance it against the force. If you measure the angle between the straight non-deformed filament and the deformed filament, then you can just basically do a balance of forces, right? To
But
determine what the tension in the string.
deformed. That is in a deformed way, right?
Yeah.
Right now, it's not deformed. I want to find out when it's not deformed. How can I find out?
Oh, I see what you're saying.
How can I get it? Very good discussion.
Can you, I think maybe what Cole was saying earlier might potentially be useful if you deform it in multiple different like magnitudes. Can you see if they like converge, I guess, to a whatever percentage of change between each deformation to calculate what it is under no tension or under no deformation?
Excellent, excellent. So let me briefly explain what I will do, and then you can see whether this fit with whether this agrees with what you are talking about, right? So our goal is to find out the rest tension. So this string has a rest tension inside. Now we want to find out this tension. Okay. So to do that, there are many ways. There are many ways to find it out. But let's see one way. So the one way to do it is we can apply a force perpendicular to it. We can apply force to it. Then this string will deform a little bit, right? It will deform a little bit, right? Now this string has a force. We apply a force to deform it. We call it F1. Okay. At this stage, the tension inside this string will change to T1. This T1 is larger or smaller than T0?
It's going to be larger than T0.
It will be larger than T0. But then what's the value of it? T1. What's its value? If I know F1, can I know what is T1?
Yeah, it's like something like F1 over sine theta t or something like that.
So we can measure this angle. Let's see, theta 1, right? So if you have an angle theta 1, then what's the relationship between T1 and F1? What's the relationship?
Is it
Mhm.
equals so okay, I'm gonna have to write it down actually because doing this in my head.
Is it the T1 sine theta 1? And times 2. Is that correct? Free
Yeah,
body.
sorry, I was thinking of solving for T1.
So F1 equal to 2T1 sine theta 1, right?
Yeah.
Okay. Now, when you pull it, you actually have here you have a small delta, right? You have a small, you define it, right? Small delta 1, right? So it deflects the string in the middle, right? So now, if you pull even further, you pull even further, you increase the force, you have F2 now, right? F2. Now, the tension here is become bigger or smaller. You have T2 now.
It's going to be a lot bigger.
A lot bigger now. You have T2 now. And then you have here you have a new delta, right? This delta is another delta. I call it delta 2 now. Let me draw it here. This one is delta 2 now. Delta 2 is a bigger or smaller than delta 1? Bigger or smaller?
It's going to be bigger.
Even bigger. So you apply force here, then that means if you apply more and more force, incrementally increasing the force, you increase the T by increasing the delta. And then this string is gradually build up the tension. So at delta 1, you have for delta 1, you have T1. Delta 2, you have T2. You have T2. By knowing how, by applying the force that you know, F1 force you can know what's the corresponding tension within the string, right? And then you can measure the delta, measure the deflection. Then you will get a series of values here, right? Is that correct?
Yeah.
Right. And then couldn't you use that to extrapolate where delta equals 0, what your tension would be?
Yes. Now, if you connect those dots together, what happens at this point? What is the value of delta here?
Tension in the resting filament.
So here, delta is 0, right? Delta is 0, but tension has some value here, right? What does that mean? What is this dot standing for? Which state?
The natural tension without deformation.
At 0 delta, right? Before you apply any force, right? Right?
Yes.
So the rest tension T0 is this value at delta equal to 0, right? So is that a good way for us to measure any string that initially carrying that carries some original tension? Is that good? Now we don't perturb we don't damage the cell. We don't perturb those anchoring point, right? Everybody follow me? Okay. This is a very simple way. Okay, so you can do that. There's other way. People also develop other way. So this, I think this way is good, but there are other way that in some the string, the tension string actually has a unique property. So if you have a string here, let's see, you have a string here. It has it is anchored. It is anchored. It has to build up some tension inside. Now, another way people usually do is they can break the string. Once it's broken, what will happen? Once this string or is tensed, the tensed string is broken, what will happen?
Once it's broken, won't the sides accelerate at a certain value and you can use F equals MA to determine what the tension was?
Not very clear to me. Can you explain in a more concise way? Once I've broken it.
So the sides will displace, and then you can measure how much it displaced over a certain period of time.
The size will displace. What is size displace?
The I would say the strings, the two sides of the actin. Sorry.
I'm not sure I'm not sure about your point, but what I can tell you what happened in reality, and then maybe that is what you talk about. Okay, we can check. So what will happen is when you break it, because this string is in a tension, then it will not just break here, it will actually retract. So this piece will go retract this way. This piece will retract this way. So then eventually this string, the two string will look like this. So there is a gap here, D. There's a big gap in between. It's not overlapped there. Okay, there's a big gap here. Do you guys understand what I'm talking about?
Yeah.
Okay. Now the size, the distance of this gap, combined with how fast this 2 point retract, how fast they retract upon dissection here, combined together gives rise to the tension force within that. Okay, so D and D dot. D dot means do the differentiation of the D with respect to the time. That means the speed of the D. Okay, these two together gives actually the D dot mostly is giving give rise to the viscoelastic property of the string. Okay, and then but the gap between that actually there's an equation involved in the polymer science can give rise based on equation, and by measuring this D, you can know what is the rest tension within this string. Okay, larger D means larger tension. Okay, so that's another way. But this method is destructive. If in a cell, if you cut actin filament, then it's not recoverable. So then that you get the tension value, but you destroy that actin, right? So that's not good. So the method initially we talked about is more reliable. Okay, but people still do this cutting because they want to prove the concepts. Sometimes they want to prove the concept that the cell actually is in the tension, then they cut it. Okay. Okay, Abigail, you understand? You understand now?
Yeah, that was the same general principle that I was going to apply, something similar to F equals ma, where if you find the instantaneous acceleration, you can relate it to the force that was applied on it at the moment that it broke.
Yeah. Okay. Okay. That's good. Yeah. So in the future, when you please talk more, and then make sure phrase your statement in a way that people can understand it easier. Okay.
Got it.
Very good. Very good. Very nice to have you participate in the discussion. Very good. Other students, do you guys can I think other students should also join our discussion, not just call question. Yeah.
So I do have a question about how the actin filament works, because I know that an actin filament is made up of lots of parts, and I believe are myosin a part of actin as well, or are they two completely separate things?
Myosin is on top of actin. They can grab the actin to stretch the actin. Okay. Now they're together. They're all together. Now what we are showing here is when we perturb the actin, myosin also will be there. We will be in a state myosin stop working there. It reached the static condition.
Okay, so in the example we just gave where we cut the actin filament, what's actually happening? Because I was kind of wondering why exactly does the actin filament retract? Is it because those myosin proteins are kind of almost coming together? Because I know that they can sort of stretch and compress, I believe.
Yeah. So good question. So in the cell, myosin is like many hands. They are pulling the actin, stretch the actin. And then once the actin is sustaining those pulling and stretching, because actin itself has mechanical resistance, so it will be stretched in a tension, and then that tension is in balance with the pulling force myosin applied. Okay. Then when we cut, when in this second experiment, when the people cut it, actually the actin will separate, will disrupt, will break, and then the tension is released. At that time, it's in a millisecond time scale. Myosin will not be able to respond that quickly to quickly generate a force or release a force. They need several seconds to respond because remember they need to take the ATP, take the energy to do the work, right? So at that millisecond moment, actually people use the high-speed camera can measure how this actin, the broken part, retract, what's their speed. And then they actually, within several milliseconds, they will rest. The broken actin will rest and then give rise to this D, give rise to that. You can finish all the experimental measurement within half a second. And then those data is sufficient for you to understand the viscoelastic property of the actin as well as the rest tension in that actin. At that time, myosin will not play the role.
Okay, interesting. That makes more sense.
Excellent. Yeah, ask those questions. Let's make the picture even more obvious. Okay, very good, very good.
I guess on that note, I have another question too. So, is there just heuristics that people are like that's widely accepted? Because I think I don't remember what the numbers were, but there's a range for the diameter that actin filaments can be, right? So they just have like some heuristic for what the elastic properties are for actin based on their diameter.
Mm-hmm. It cannot be stretched too much. It has its limitations. Okay, so that's actually why the last time I think Zeyuan asked the question that whether we can measure the tension by measuring the diameter change of actin. That change exists, but very tiny. It is way above the resolution of the microscope. Okay, so if you want to do those experiments, you need because it's just a sub-nanometer change, so it's very hard to measure by optical microscope. Okay, so using the first method we talk about here, by perturbing the filament and then do the extrapolation, measure the rest tension, is more convenient. Okay, and in the next slide, I will show you a method how to apply this F force to calculate the T.
Okay, I guess what I'm curious about is, so are they just taking, someone's done, like, measured the modulus of actin filaments and knows like the mechanical properties of them, and they assume that when they're calculating the tension after cutting it?
Yes, you can do that. Yes. Good. Okay, other students, let me see. Many other students have not asked questions before. Do you guys hear me? Like Yibo, Zhihong, Renjing, can you guys hear me? Do you guys hear our discussion?
Yes, I can hear. Yes.
Okay, excellent. Please join the discussion. Not only listening is useful, but join the discussion, have the brainstorm of the idea is much more encouraged. Okay, that's how we all learn and find out our confusion, clarify our confusion, clarify any trouble there. Okay, good, very good. Okay, let's continue. Okay, so to apply the force inside the cell, actually we have developed one method. There are other methods as well, like what Jason said about the optical tweezers, right? There's another method one of my students developed. So he actually can inject nano magnetic beads into the cell in a very non-invasive way, and then applying the magnet to control the magnetic particle. Those are black dots. And then he can apply really precise force to deform the cell, any organelle in the cell, including the nuclear, as well as bend the actin filaments. Okay, so here are two examples. And then by looking into the chemical signal in the cell through some imaging ways, he can directly determine the relationship of the mechanical perturbation and the cellular signals. Okay, so this is actually one way to non-invasively apply a force within the cell. Actually, he can also do it within the nucleus, okay, using the magnetic field. The optical tweezer people mentioned is also a very useful method. However, optical tweezer, the force magnitude cannot be too large, because it is about 10 to 100, can be 10 to 100 times lower than the force applied here. Okay, so sometimes it is optical tweezer is preferred to deform single molecule, but not to deform those single actin filament or the nuclear. The organelle in the cell is not very capable for the optical tweezer. Okay, and also the student can also, after the experiment, he can take out those magnetic particles to avoid any further perturbation to the cell. So this actually is a very unique method, very convenient to use for the cell mechanics or organelle mechanics study. Okay, good. So that's all the brief.
Can I ask, just out of curiosity, how they take them out? I'm just curious how they would take it out without destroying anything else in the cell.
Yeah, actually, we can use some very tiny micropipette, glass micropipette, to insert into the cell and then suck this particle out. Because the micropipette is so small, when they penetrate the membrane and retract from the membrane will not get damaged. They will heal itself. So then it's no perturbation to the cell.
So they're like in the below 10 micron size?
Yeah, actually can be even smaller. Can be hundreds of nanometers. Yeah.
Oh wow.
Can do that. It's a very delicate experiment, but it's all doable. Okay. Yeah. Good question. So that's all about about discussion about the cytoskeleton. So I mentioned that two components are very important in the cell in terms of the mechanical properties. One is the cytoskeleton. Another one actually is the membrane, the cell membrane. So in the cell, is the organelle and the molecule and protein that actually are very, very crowded in the cell. They're very crowded. To make this, to make sure those crowded environment will not perturb cell normal function, nature actually developed a way to use the membrane that is made of the lipid. Okay, use the membrane to compartmentalize the cell. So in the cell, there are actually many different sections. That is, all those sections are enclosed by the membrane, and within each compartment, they do their own work without perturbing the other parts. Okay, so cell nuclear is one part. Cell nuclear has the membrane, and then this ER also contains membrane. Those pink entities actually are the mitochondria. They are also enclosed by the membrane, and the whole cell itself also enclosed by membrane, so that they are not perturbing neighboring cells. Okay, so membrane is actually a very good barrier used by the cell to separate the work, so leave a different job to be done in an independent way. Okay, this actually protect the cells, especially the eukaryotic cell. For the prokaryotic cells, as we mentioned before, they don't have those membrane compartmentalization because they are very primitive, and within that small space, because their job also are simple, their function also much simpler. Okay? So in the cell, here is actually a picture of different organelle. They are all enclosed by the membrane, like the lysosome. Lysosome actually is the one that doing the recycling of the protein. Okay? So they are all enclosed by the membrane. This one here, the text is the block. This actually is the nucleus. This one is the nucleus. This one is the mitochondrial. This one is the Golgi apparatus. The next two are the ER. One is a smooth ER, smooth endoplastic reticulum. Another one is a rough endoplasmic reticulum. Okay, both are very important. For the rough one, it actually looks this smooth and rough is based on their morphology. For the rough one, they are rough because they are embedded with many ribosome organelle, because this ER is the position that the translation is taking place. So many ribosomes inside. They're all doing different work to generate different proteins, and then those different proteins will be trafficking to different location of the cell to perform different functions. Okay, so the cell membrane actually is compartmentalize all those different organelles to let them perform distinct tasks. Finally, the whole cell is covered by a membrane. Okay, the bilipid membrane. This membrane actually is very thin. So here the scale by is 30 nanometer, and the cell membrane actually is very thin. It's about 5 to 10 nanometer. 5 to 10 nanometer. Okay. Now here, if you look at this membrane, this is all SEM picture. SEM electronic microscope. Okay. So if you look at this membrane, you may notice this membrane looks very uniform, right? However, in reality, this membrane is not very uniform. I give a range there, 5 to 10 nanometer, right? 5 to 10 nanometer actually is not very uniform. I'm not giving it a single value. I'm not saying it's a 7.5 nanometer. Okay, I said it's a 5 to 10 nanometer. Okay, so it's a range. That actually is very interesting because of the thickness of the membrane can vary in different thickness. In fact, on the membrane, many different proteins they can take advantage of this thickness difference to cluster together. Some of the protein, their size is within the range of 5 to 6 nanometer. they actually preferably will travel to the thin membrane region, and then they form a patch. They will form a patch. Some other protein that in the range of 8 to 10 nanometer, they actually prefer to go to the thicker region of the membrane. So if you use the fluorescent light to label those proteins together, label the transmembrane protein with different color, like red and green, you actually, if you look at the cell, you will actually see many patches. And then green may corresponding to the thin membrane and a small protein. Red corresponding thick membrane and bigger, a little bit bigger transmembrane protein. Okay, so that's very interesting. So that's actually a very active area people are studying. Is it because the protein change the membrane thickness, or is because of the membrane thickness regulate those protein to express on them? That is actually a very active area to study. Okay. Now, as I said, this membrane is made of the lipid phospholipids. Okay, so those phospholipids actually is the isolator. They do not allow. They actually contains the hydrophobic region inside. Hydrophobic means that water cannot go through that. Okay, so that means if a cell only contains those phospholipids on the membrane, then the water cannot be exchanged. Water cannot get into the cell that easily. So nature actually solved that problem by embedding many proteins, many water permeating proteins on the membrane. So membrane is not purely membrane. It actually contains many transmembrane proteins, like those proteins. Okay, those proteins. Can you guys hear me?
Yeah.
Yeah. Okay.
Yes.
Those proteins allows water, solution, and small molecules to go through the membrane, so that the cell gain the information change capability to their external environment. Okay, and furthermore, those proteins actually, many of them help cell to anchor to the extracellular environment, and the intracellular environment in the intracellular region, those proteins actually anchor to the cytoskeleton. They anchor to the cytoskeleton. Okay, so this actually causes a problem to the cell. If we have pure membrane, pure lipid, so those are lipid. We will talk more about lipid in the next few lectures, but here I just want to give you an overall sense. So here is the lipid, bilipid membrane. If we only have pure lipid membrane, if we perturb it, they actually are very fluid-like. If you apply some force to the lipid, they will quickly diffuse. The force will quickly diffuse to distant region. But in reality, in the cell, there are many of those proteins embedded there. And then those proteins anchored to the cytoskeleton. And as we said, cytoskeleton carry the force, like carry the tension and compression force. So those cytoskeleton actually anchor to those transmembrane protein. That makes the propagation of the force on the membrane to be inhibited. So the deformation of those lipids cannot propagate very long distance, because once they propagate, they will encounter those blocks embedded in that, those block the protein there, and then those proteins are carried by is anchored to the cytoskeleton, so they are very rigid. They cannot move that easily. So the force carried by those the lipid will bump to the protein and then dissipated there. Mm-hmm. This actually can be calculated by the equation. So people develop an equation. Here, V, in the left-hand side, V is the speed of lipid propagation. If you apply some force to it, lipid propagate, the speed is called V. Phi here, this thing is actually the force, Delta is the gradient. The delta phi is actually gradient of force. If the force has a gradient, that means there's an imbalance, right? So then the the lipid will gain energy, and then they can move. And then when they move, they can get a velocity v, right? So this is talking about the force induce a velocity. But this velocity actually also regulated by another two parameters. One is a k. This k actually is a value called permeability. It's called, let me see, it's a Darcy permeability. K is Darcy permeability. It actually can be described. It can be described by another equation, and that equation contains mainly two parameters. Those parameters are given rise by those embedded proteins. One is A. A is the radius of the protein. So you have many proteins here. Their average radius actually is contributing to the K. Another one is called phi. Phi is the area fraction of those embedded protein with respect to the whole surface of the membrane. If you have many of those transmembrane protein there, then this phi will increase. Means there are many transmembrane protein anchored. Okay, so basically this equation can give rise, can describe the K. And then another one is the viscosity of the membrane. Okay, viscosity. So higher viscosity also give a lower velocity. Okay, so K and this K actually is coming not from the theory. It's mostly coming from the experimental measurement, experimental derivation. Okay? Basically here, what it talks about is the force applied to the membrane and the membrane proteins concentration, distribution, area, size, as well as the inherent property of the membrane. All three of them collectively contribute to how fast membrane can propagate. Okay, and this membrane propagation is another way, in addition to the tension propagation in the cytoskeleton, to give the signal to the cell. So if you perturb the cell in one location, the membrane can also serve as another avenue to transmit the signal. Okay, but in reality, because of these embedded proteins there, and this equation actually shows that if you put the number in, this velocity actually is very small value. Most of the signal transmitted, mechanical signal transmitted actually are through the cytoskeleton, because there are no perturbation, no inhibitions around the tense cytoskeleton. They are very efficient in deliver the mechanical signal. Okay. Here, yeah. So if you think about the membrane, it's more like a gel. Okay. So membrane is like a gel, but within it there are many protein embedded, like the fluids, many fluids embedded there. So if you perturb the gel in one location, the deformation of the gel cannot move that easy, because there are so many fluids that those anchored protein embedded there. They can dissipate the energy, dissipate the force there. If you have a pure gel here, then you perturb the gel in one end, then the deformation can be very easily seen in another end, right? But now you have a lot of fluids embedded, then it's not that easy. So the membrane deformation is not the major way the mechanical signal transmit within the cell. It's mainly through, the signal is mainly through another way, like a cytoskeleton is one way. Cytoskeleton is one quick way. Okay. So here is another video to give you the impression how deformation of membrane looks like. So this actually is a cell extend a dendrites towards outside. Okay, and then people use the optical tweezer to stretch the membrane, and then you can see how fluid the membrane looks like. So here is the optical tweezer, and then it can go to bound the membrane and then stretch the membrane, and then shear the membrane. Now it pulls the membrane now, stretch the membrane. Okay, so you can see this is a small lipid. It is stretched. Okay, and then you can see how fluid they are. But in fact, in this case, there are not many embedded protein there, because this is a newly formed dendrites. Okay, so the membrane itself is a very fluid. Okay, good. So so in summary, the cell membrane as a second important organelle we emphasize here. It actually has the essential role to help cell to maintain a constant internal environment, and even inside the cell, it helps cell to maintain the compartmentalization and let different organelle perform distinct functions. And also because of those transmembrane proteins, membrane actually serves as a selective permeable barrier, so that it can perform the chemical, mechanical, and electrical functions. For chemical, membrane actually is an interface for the cell that the information is received from the adjacent cell and the extracellular signals, because those transmembrane proteins actually can selectively let the ion, different element, different ion to go through. So then they can chemically communicate with the neighboring environment. Okay, mechanically, even although we know that on the pure membrane, that cell membrane, that the mechanical propagation is small, but but in that local region, the ion channel and the cell adhesion molecule there still are influenced by the mechanical property of the membrane. Okay? And also eventually for the cells that are electrically rich, such as the neuron and the cardiomyocyte, when the electrical charge on the membrane get disturbed, then they can generate an action potential, and then those action potential can propagate on the membrane, and then they can go in a long distance to deliver the message. So membrane not only serve as an interface, important interface, selective permeable interface and barrier for the cell to maintain their function, but also can use the chemical, mechanical, and electrical way to communicate with the neighbors, with the external environment. So it's very important. Okay, right now it's 3:53, and then we can continue our lecture in the Wednesday, and then we will talk about some really useful calculations first. Okay, good, very good.
Thank you.
I have a